Hello everyone. Good morning, good afternoon, and good evening, depending on the place you have joined us from. I would like to welcome you all for today's webinar on gut wellness and its role in immunity organized by FIP. My name is Bidhata Khatri and I am the moderator for today's webinar. Currently, I serve as FIP Health and Medicines Information Section Secretary, and I graduated with a Doctor of Pharmacy from Acharya and B.M. Reddy College of Pharmacy in 2017. Currently, I work as Senior Drug Safety Associate at a CRO in India. Some of the key announcements regarding today's webinar. This webinar is being recorded and live streamed via YouTube. The recording will be available on our website events.fip.org. You may ask questions using the question box provided. You are welcome to provide feedback to webinars@fip.org. If you want to become a member of FIP, you can visit the website of FIP and register as a member. For those of you who want to listen to the Portuguese and Spanish translation, you can join the respective channels. FIP would like to thank Sanofi for supporting this online event through unrestricted funds. So this is the general overview of today's webinar. It's my honor to introduce some of the distinguished panelists today for the webinar. We have 4 panelists who will be joining us later. So today's program, first panelist is Saman Khalesi, who will not be able to join us live. However, we will be playing his recorded presentation. Then second presentation will be by Anna Faria, and she will be presenting about disease to therapeutic response. Then we have Ender Pelivanoglu, who will be presenting about use of probiotics to correct dysbiosis of normal microbiota. Then we have Metehan Ojen, who will be presenting about prevention of respiratory tract infections by modulating gut balance. Then we have questions and answers, and finally, closing remarks. So the learning objectives of today's event would be understand dysbiosis and gut wellness, understand the role of diet in immunologic modulation, and then understand the use of probiotics for gut wellness. So the first presentation will be by Saman Khalesi, and it is a recorded presentation. He's a senior lecturer and discipline lead nutrition at Central Queensland University. And his bio, we have a bio of him. He will be presenting through recorded presentation. So just a minute. So he's a postdoctoral fellow of the National Heart Foundation of Australia and a senior lecturer in nutrition at CQUniversity, Australia. He has more than 56 peer-reviewed publications with more than 1,500 citations. He has led or involved in competitive external and internal research funds and industry collaborations and supervised PhD, masters, and honors students. The central theme of his research is to build the capacity of consumers to make informed food choices. His SALT Aid project aims to develop a low-cost, highly accessible, and personalized 24/7 online coaching program to reduce salt intake to prevent and manage hypertension. Also, together with his collaborators, he investigates the role of lifestyle and gut health in our mental and physical health. So next we will be watching his recorded presentation.
Hello, everyone. My name is Saman Khalesi. I'm a postdoctoral fellow of National Heart Foundation and a discipline lead in nutrition at Central Queensland University. It's my pleasure to have this talk today. I couldn't attend the live session, but my email address is here, and I'm happy to answer any question related to my talk or even share my slides. So, for a long time, the term bacteria was only associated with infection and diseases. But nowadays, we hear a lot about healthy bacteria, probiotics, gut flora, and so many other terms that they are referring to the beneficial microorganisms of the gut. Just a quick review about the history of gut health. Mishnikov was probably the first person that pointed out the benefits of some of these bacteria, and that was around 1900. But it was only in 2001 that WHO officially defined probiotics. And now we know that we have more than 40 trillion microbes living in or on our body, and given that on average we are around less than 30 trillion human cell, we are almost half human and half microbe. I'm pretty sure you know what the definitions of probiotics, prebiotics, and symbiotics are, but since the discovery, they have been increasingly popular, with many studies trying to find the links between gut health and our health and diseases. And right now, if you search probiotics in PubMed, you will have more than 37,000 articles just on one term on probiotics. And because of all these health claims, we can see a drastic increase in the number of food sources and supplements that they claim to have probiotics. In Australia and New Zealand only, we have seen a 200% increase in the number of probiotics products in only 3 years. And I'm sure this is the same or more in any other country. Okay, so these are just a list of very well-known probiotics and prebiotics and natural resources. But obviously, we have a lot more, and we keep updating these, increasing the, updating this list, and this list kept getting larger as we discover new probiotic bacteria and microorganisms. So, the health of our gut plays an important role in our physical and mental health. And our microbiome symbiosis define this health of our gut. And we have this, and the symbiosis is when we have a balance in the diversity and the number of helpful and commensal bacteria compared to pathogenic bacteria. And a healthy gut can boost our immune system because of the role that they play as a first line of defense against harmful bacteria. They remove the food and food from the harmful bacteria, and they also produce bacteriocins that they can kill harmful bacteria. And they also educate our immune system to understand which bacteria is helpful and which one is harmful. But it is not just the presence of certain bacteria that can lead to health issue. Even the absence of some or all of our bacteria can lead to health issues. And we have seen this in when broad spectrum antibiotics are used for an infection or disease, which kills normal gut flora and leading to antibiotic associated diarrhea. Now, there is also a link between our gut and brain. That term's gut-brain axis, and that's referred to the bidirectional communication that happens between our GI system and our central nervous system. They talk with each other through the byproducts, the neurotransmitters, hormones, and metabolites that they have. And also, we have seen an increase in pro-inflammatory cytokines and interleukins as a response to the unhealthy gut, which has been linked to depression and anxiety as well. And there's also evidence that our gut flora can regulate the circadian rhythm by producing by increasing the production of tryptophan amino acids, which can be converted to the sleep promoting hormone serotonin and melatonin in our system. And on the other side of the equation, a stressful life and anxiety can also change the composition of our gut flora too. Um, so We do know that our gut bacteria contribute to the metabolism of the food as well, and they have enzymes that we don't have, and they can break down some of our polysaccharides and dietary fibers and polyphenols, and ferment those polysaccharides to produce some short-chain fatty acids for us, like acetate and butyrate. And butyrates are fuel for our colon cells, and they also have anti-colonic cancer properties. With our gut bacteria can also produce some vitamins, especially vitamin K and B vitamins, and those, and technically almost 1/3 of the vitamin K that we have or we need are produced by our gut bacteria. Again, on the other side of the equation, if we have a poor diet, like high consumption of saturated fats and low consumption of fruits and vegetable or dietary fiber, and high alcohol intake, they can affect our gut balance and disrupt that equilibrium as well. They will also help us with the prevention and management of chronic diseases. And I will talk about some of our research funding in this area soon. But the anti-inflammatory characteristics, the lipid-lowering potentials, the enzymes and the byproducts that they have, they can help us with the antioxidant, with having a more antioxidant pool in our body and help with the prevention and management of diseases and inflammations. Just an example, the lignans, which are polyphenol components found in flaxseed and some vegetables and some legumes, they are a potent antioxidant, and they can have yeah, they have cardiovascular protective effect as well, but they need to be activated in our system and converted to interleukin to have that antioxidant potential. And this conversion and activation happens by our gut bacteria for that matter. And we also have some bacteria like oxalobacterium that they can regulate and metabolize oxalic acid in some vegetables as well as coffee, and prevent the formation of kidney stones in those who have calcium oxalate stones. And so it is important to note that this is still a very new area, and we still don't know much, but we know that the effect of bacteria is a strain specific, and it depends on the dose and the duration of supplementation and the health status of people. Also, it's important to note that viability of these bacteria and how effective they can be in, or supplementations can be in surviving the low acidity of a stomach is another debatable issue. So let's talk about some of our research and some of the different findings that we had. So in one of our very first study, we looked at the effect of probiotics on, or the effect of a specific strain of probiotics, which was Lactobacillus casei, in reducing bioavailability and absorption of aflatoxin. Aflatoxin are potent mycotoxins that found in some foods like pistachio and corn, and we saw that this strain of probiotics can help reducing the absorption of aflatoxin in body. But this was an animal study, and this was pretty much the only animal studies we did. Then we moved into human study, and here I'm going to present some of the results of our systematic review and meta-analysis of randomized controlled trials. We know that systematic reviews are on the top of the hierarchy of evidence, and we use the result of these systematic reviews to develop clinical guidelines. So the very first one of the very first systematic reviews that we did was looking at the effect of probiotic supplementation on liver function. We know that pathogenic bacteria can produce and release endotoxins such as lipopolysaccharides and hepatotoxins, and they can cause inflammation of the liver. In this meta-analysis, we found that probiotics and symbiotics can improve liver function tests, especially in those who have liver disease or when supplementation is longer than 8 weeks. And here are some of those bacteria that were more frequently studied for their effect in this area. In another study, we found that some probiotics can improve glucose intolerance and insulin resistance, especially in those who have a baseline higher fasting blood glucose, or when multiple species of probiotics were used. And these are again a few other probiotics that were more commonly studied in this area. And just to note that some of these studies had supplementations that use multiple species, and some they had supplementation that used just one species of bacteria. So in those studies that use multiple species of bacteria, it is a little bit difficult to note if that effect was specific to one bacteria or not. And in one of our actually highest cited systematic review, we found that probiotic supplementation, especially in those with baseline higher blood pressure, and when supplementation is longer than 8 weeks, can lower blood pressure. Some bacteria produce angiotensin converting enzyme inhibitors, sorry, that can inhibit the production of angiotensin 2, and it can reduce blood pressure. And these are some of the well-known bacteria that were studies in this area. We also found that they are effective in improving lipid profile if they are consuming more than 8 weeks, especially if they have a higher baseline, triglyceride, cholesterol, and LDL lipids. And we found a similar effect on subjective measure of sleep following probiotic supplementation, which suggests that they may help with the reducing the stress, inducing glycocorticoids, and improving sleep. And these are some of those bacteria that may help with the production of GABA, or improve the absorption of tryptophan and sleep hormones. But again, they are more effective if supplementation is for more than 8 weeks. So you can say that there is a trend here that appear, but about the probiotic supplementation duration, but this number is not an absolute one. It is just because it takes time for probiotics to be able to change the gut and to colonize, and we believe that at least 8 weeks of supplementation is required for some of those to happen. And we have done a number of RCTs for the effect of probiotic supplementation on migraine characteristics as well as alcohol metabolism. I'm happy to share those for anyone who wants to, but until further studies that in this area, we actually can't really draw a conclusion on those findings. So as you can see, most of our studies showed beneficial effects in people with health conditions. So we wanted to see if there is an evidence for healthy adults, because majority of supplement manufacturers actually actively target healthy adults. Well, our review of healthy adults and probiotic supplementation suggested that probiotics can provide transient improvement in gut concentration of supplement-specific bacteria. And well, some studies suggested that even the passage of bacteria through our gut can have health effects, but the evidence didn't suggest a permanent change in the gut colonization in healthy adults. But we could see an improvement in immune function of healthy adults, especially in response to the common cold with some bacteria like Lactobacillus casei, Platinum, and Bulgaricus. But the evidence was again inconclusive for the benefit of probiotics in lipid profile and cardiovascular risk in healthy adults. We also saw that probiotic supplementation in healthy adults may have some benefits for gastrointestinal discomfort. So in overall, for healthy adults, it may help with the they may help actually with the immune response and the GI discomfort, but no evidence of the beneficial effect in lipid profile or the ability to cause persistent change in gut flora of healthy adults. So Well, all of these are very interesting findings, but to translate these findings into impact, we need to know if people know about these bacteria and they consume these ones, and if they consume probiotics, who is the characteristic of people who are consuming them. So we surveyed more than 1,200 Australian adults, and one of the interesting findings we had was that most people who were consuming probiotics were women who were mostly healthy and who were health conscious and consumed these probiotics mostly to maintain health. Most probably not the one who needed these products or should consuming these products based on our research. And our males and those who are unhealthier, who could really benefit from probiotics, were not really using them. But the good news was that they were willing to use these products if the health professionals recommend them. So maybe our take-home message is the policies and interventions to improve the knowledge of gut health and probiotics in public, as well as our health professionals who are willing and updating themselves around this area and willing to provide recommendation of probiotic use for those who really benefit from these products could actually improve gut health of our population. So it feels like we are on the right path with having all of these seminars and talks, but we still need to do a lot more to get to the point that we have a healthy gut in our population. Thanks a lot for listening.
Thank you, Dr. Saman, for your wonderful presentation. If anyone has questions regarding his presentation, they can email him and he will be replying through email since he is unable to join us live today. So the next panelist for today's webinar is Ana Faria. She is assistant professor at Nova Medical School, Universidade Nova de Lisboa in Portugal. And Ana Faria graduated in biochemistry from Faculty of Science at University of Porto, and she completed her PhD in chemistry in 2010 at the same university. And then she had aggregation title in 2022 and currently is assistant professor at Nova Medical School, Universidade Nova de Lisboa. She is a researcher at Comprehensive Health Research Center and has published more than 70 papers in scientific international journals. She has supervised master and PhD students and has been involved in more than 15 financed projects from national and international entities. Her research interests are food bioactives and their impact on human health, namely through nutrient transport modulation, impact on microbiota composition and functional activity. Over to you, Anna.
Thank you very much for the presentation, and good afternoon to you all that are joining us today. My presentation regards the role of microbiota and host interaction in disease and in therapeutics. So I'm starting by presenting some curiosities about microbiota, and we all know that this community of microorganisms colonized our body, and if we put them in line, we can circle 2.5 times the Earth. So we have much more cells from bacteria than cells of our own. So this is a huge community that is going to influence our health. And we know that our health is influenced by several factors: age, physical activity, medication, environment. But also microbiota has gained a relevant importance, since microbiota can also influence our inflammatory, oxidative stress and metabolism pathways that are going to implicate our health. And we know a bit more about microbiota and the interaction of microbiota metabolism and the host metabolism, and we know that they can interact differently and within different pathways. We have the dietary fibers and the production of short-chain fatty acids, the secondary biliary acids that are going also to implicate GLP-1 or PYY and have an effect on satiety, fat storage, adipose tissue inflammation. We also have now the characteristic, the trimethylamine, that is a product characteristic from specific bacteria, from phosphatidylcholine and L-carnitine, that is oxidized in the liver and is a risk factor for cardiovascular disease, and also several other molecules that can interact with the host and implicate in host health. So this is not new, and we know we have some of these mechanisms well described, but we know that our microbiota and our microbiota composition is going to influence all these metabolites and the formation of these metabolites with impact in human health. We know that we want our microbiota in a homeostatic way, but most of the times in disease we have a dysbiotic microbiota that can translate in a decrease in beneficial microbes, an increase in pathogenic microbes, or even a reduction in diversity. So we want to search for strategies to maintain our microbiota in a homeostatic way. But not only the microbiota environment can interact with the host, but also the environment of the host can influence our microbiota. And we know now that the metabolism of our colonocytes can influence the amount of available O2, and the obligate anaerobic bacteria have difficulty in growing in this high environment with O2. So if our colonocytes are not properly function and our mitochondria are not properly function, we have available O2 that is going to cross the cells and here increase the O2 pressure and modulating our microbiota. So the host environment influences also our microbiota. And we know that a dysbiotic microbiota, particularly in the first years of life, is going to be implicated with a gut dysbiosis in adulthood and health outcomes in this adulthood. And we know that a dysbiotic gut microbiota in early life is going to be implicated with several diseases such as obesity, diabetes, allergic disease, and others. So we have here a window of opportunity to modulate and to take care of our microbiota in this early phase of our life. That is going to be very important for the development of immune and endocrine system, and to be and contribute to a healthy state in the adulthood. And we have several, we know now that we have several strategies to modulate our microbiota. We've just seen prebiotic, probiotic, and symbiotic as strategies to modulate our microbiota, but we also have exercise, nutrition, and in another way, the fecal microbiota transplantation. And these are all interventions that can be made to improve our microbiota composition and to improve microbiota function. We know now that several factors impact gut microbiome, and now I'm focusing here in this early life, the infant feeding method and the birthing process, but particularly in the infant feeding method. And to study this, we conducted a study with preterm infants that were born in a Portuguese maternity. And this preterm infant, we collected the meconium, the first fecal sample, and they were fed for 26 days with mother on milk, donor on milk, or formula, donor human milk, sorry, or formula. And after 26 days, we collected a sample of feces again. And with this design, we observed that mother on milk had induced a different composition of microbiota than donor human milk and formula. And we have a higher richness and a higher richness at phyllo and at genus level in these babies that were fed with mother-own milk. Instead of donor human milk or formula, in which the diversity was a bit lower. Also, we saw that this bacterial richness was increased in these babies that were fed with mother-own milk, and that this diversity was also a tendency to be increased in this group, and there were decrease in the fecal alkaline phosphatase activity in babies that were fed with formula. And alkaline phosphatase is here a marker for intestinal maturity. But we using the same cohort of babies, we also saw that the maternal bacteria can influence the baby microbiota, and the maternal bacteria from gut and vagina can play a role in shaping the neonate's gut microbiota. And we saw that the babies that were born extremely preterm, which means under 28 weeks, had a microbiota that were more similar to vaginal microbiota than babies that were born after 28 weeks. So mother to infant bacterial transmission or influence and modulation is controlled and is a time-specific process. But we also know that diet is one of the easiest ways to modulate our microbiota with impact on our health. So we conducted several studies with nutritional interventions, since we know that diet low in animal fat and protein but high in fiber can induce a specific microbiota that is a short chain fatty acid producer that is going to impact our metabolism, particularly our glucose metabolism. And we also know that diet high in animal fat and protein but low in plant fiber is going to modulate a different microbiota that can produce other kind of metabolites with impact in host health. So we conducted a study to understand the effect of a fermented beverage, in case beer. Beer is a rich source of phytochemicals, some fiber, but vitamins and minerals. And we conducted a double-blind randomized two-arm parallel group trial in which our participants drank during 4 weeks 330 milliliters of beer daily, and we collected samples before and after this intervention. And these two groups, they drank beer with alcohol and beer without alcohol. And what we saw was that in both groups, after 4 weeks drinking 1 bottle of beer a day, there were an increase in microbiota diversity, and it was independent of the alcohol level of the beer. Also, we have a tendency here to increase alkaline phosphatase activity. That is also an intestinal barrier function marker. But we know that we can modulate microbiota from healthy subjects with dietary intervention that we just saw, but we also know that some diseases and some metabolic diseases have an influence of microbiota in the pathophysiology of the disease. And we know that for type 2 diabetes, we have risk factors that are non-modifiable, but we have risk factors that are modifiable, and gut microbiota is one of them. And we now have several scientific evidence that relates some types of microorganisms with an impact in type 2 diabetes. And we have bacteria that are positively correlated with the disease and some bacteria that are negatively related with the disease, and also the metabolites that are produced by these bacteria. So, taking this into account, we conducted a study in type 2 diabetes individuals that were included in this study, and they had an interventional nutritional intervention during 12 weeks. And this nutritional intervention was based on the Portuguese Mediterranean food wheel, which means a Mediterranean pattern diet. And we have here the results. After 12 weeks, our participants increased their adherence to Mediterranean diets, and glycated hemoglobin decreased after 12 weeks, and this was a significant decrease. Also, HOMA-IR, a measure of insulin resistance, was also decreased after 12 weeks. And here we saw that the bacterial richness was increased with this nutritional intervention, and the ratio of firmicutes bacteroidetes was decreased, and these parameters had a clinical relevance. Also, we correlate the microbiota diversity with glycated hemoglobin, and this was a negative correlation, and also a negative correlation between richness and fasting glucose and HOMA-IR. So this strengthens the role of a dietary intervention in type 2 diabetes management, improving metabolic control of these individuals. But we have this as an homeostatic microbiota, and this is what we want. But sometimes we have a dysbiotic microbiota that is related with the different diseases, and now we know that this dysbiotic microbiota is associated with the disease, but not only associated with the disease, but also with the severity of the disease or the prediction of the disease. And for type 2 diabetes, we now know that some microbiota profile is related with risk of type 2 diabetes. And also for COVID-19, we know that dysbiotic microbiota is related with hypertension, cardiovascular disease, obesity, that are risk factors for the severity of COVID-19, and these conditions are related with a dysbiotic microbiota. So we can use microbiota not only to predict the disease, but only to predict the severity of the disease. And this was a study that we conducted in our first wave of COVID-19 in Portugal. And we analyzed microbiota from patients with COVID-19 with different severity of the disease. And we saw that a low diversity of microbiota was associated with a high severity of COVID-19. And this can also be perceived in these trees here in the right side. And also we separate our participants by their location. Obviously in the ambulatory we have the less severe patients, and ward and ICU we have the most severe patients. And as you can see here at phyllo level, we have an increase in proteobacteria in patients that were in ward or in ICU, which were patients with a severe form of the disease. So we could construct a statistical model that relates the diversity of microbiota with COVID-19 severity in order to predict the disease severity. And here I introduce another topic that are rheumatic disease. And rheumatic disease are chronic inflammatory disease that have a component of microbiome. And this component is different in the different rheumatic disease. But we know that for ankylosing spondylitis, which is a kind of rheumatic disease, we have a different taxonomy of bacteria in patients with ankylosing spondylitis and healthy control. And this is here in this graph, in the E graph, we have the ankylosing spondylitis patients with a different composition than healthy controls. So we know that the microbiota composition in this disease is different, but also that the metabolites that are formed from this microbiota are also different. And in this paper, the authors saw that the tryptophan metabolites, indole, 3-acetate, and 3-acetaldehyde were different between healthy controls and spondylitis, ankylosing spondylitis patients. So we know that this is a chronic inflammatory disease, affects young people, mostly men, causes pain, fatigue, stiffness, is a condition that takes a lot of quality of life. But if we early diagnosis and have an effective therapy, we can increase this quality of life. And now the second line of therapy is a biotechnology therapy that is used. It's an anti-TNF therapy, but only 60% of these patients respond to this therapy, and 40% don't respond to this therapy. And this therapy is expensive and has a lot of secondary effects. So we are now conducting this project, and this is just a project yet, that we intended to use microbiota as a marker for the response or non-response of these patients to this anti-TNF therapy. So we intended to identify specific profiles of oral and gut microbiota that can predict us the response of patients to this therapy in order to base their treatment and specific personalized this treatment for these patients. We have some preliminary results, and we included patients before they start this therapeutic intervention and after their clinical evaluation after 14 weeks. And this is the preliminary results from gut microbiota, and we see here that we have the non-responder patients, the ones that didn't respond clinically to the therapy, and the responder patients before the treatment. And as you can see, this is a very different profile from non-responders to responders. After 14 weeks, this profile tended to be similar, despite these patients didn't respond. And here we have beta diversity that is also demonstrates this difference between the beginning and the end of non-responders and responders patients. This is just the phylocharacterization and is clearly different. The initial microbiota of patients that are not going to respond is very different from patients that are responding to the treatment. So we know that all the disease begins in the gut and that microbes have the last word. So I just want to thank for the finance of these institutions and thank my team here in Nova Medical School. Thank you very much.
Thank you, Anna, for your wonderful presentation. Our next panelist for today's webinar is Dr. Ender Pehlivanoglu. It's quite a difficult pronunciation. Sorry if I mispronounced it. He is Professor of Pediatric Gastroenterology and Nutrition at Istanbul Kent University in Turkey. Ender Pehlivanoglu, MD, is a professor of pediatric gastroenterology, hepatology, and nutrition. Doctor has current research interests focused on the digestive diseases, Helicobacter pylori infection, gut microbiota, celiac disease, and food allergy. He is a member of European Society of Gastroenterology and various other societies. Over to you, Doctor.
Well, thank you for the presentation. You did very well. Even the pronunciation was okay, don't worry. Dear audience, my task is to talk about, Well, I hope you have been hearing well. So my task is to summarize dysbiosis and the role of probiotics in certain disorders, which are important in health and diseases as well. So the gut is sterile at birth, as you know, not at all nowadays. We realize the importance of the intrauterine microbiota. So there are some other sources of microbiota as well. In case of disruption of the ecosystem, some health issues may occur. These are necrotizing enterocolitis, asthma, allergy, atopic dermatitis, diarrheal illnesses, IBS, and I'm going to mention more about constipation as well. So the microbes are available in the body, as you know. In different parts of the body, they have been colonized to provide us health and some other beneficial functions. As I mentioned you, infants require the microbes during at birth, but there are some microbiota which are present even in placenta, and they have some functions during intrauterine life. Of course, the placental microbiota is very important to trigger immune response in the fetus. It programs immune development of the fetus. That's why, in addition to this intrauterine population, the type of delivery is also important. So the colon microbes originated from these sources exert their beneficial effect, which are important in many functions. So it seems this has been well, there are many positive and negative effects of the colonic microbiota, which I am not going to summarize. I think there is a kind of internet connection problem as well we are facing. So these microbes improve the immune system and vitamin synthesis and pathogen suppression and some magical products such as short-chain fatty acids. I will touch very briefly. So the secret of the life is maintaining the balance, which is called eubiosis. In this slide, I would like to emphasize the importance of this balance. And if I ask you the question, which bird is free? Probably majority of you will answer none of them, because the freedom depends upon the other one's freedom or the healthy status. So that secret, eubiosis, is important, and if it's not well maintained, there will be failure of host microbiota balance, and eventually will lead alterations of gastrointestinal microbiota, which is going to lead dysbiosis and then enteropathogenic syndromes. So dysbiosis, let's define it, is a microbial imbalance or maladaptation on or inside the body. So this is not specific for gastrointestinal tract. Dysbiotic health issues are IBS, IBD, infections, food intolerance, obesity, cancer, liver disease, pancreatic disorders, neurological problems, including Parkinson and Alzheimer. Probably this kind of approach is quite new in medicine. In conventional medicine, we have learned that the brain is in the skull, but that's not the reality. So, interaction between is by microbiota, of course. The interaction is important, it's crucial. It's crucial to realize may I ask Ruben or Mila to help me?
I can hear you, Professor.
How to move forward? You can do it maybe, it's not functional anymore. Okay, please do it instead of me if it's not working properly. But what I was trying to tell the audience that the interaction is important between the skull, the brain in the skull, and the enteric system, which is located in the belly. That's why the genius Salvador Dali has pictured this event very well. So anyhow, let's move forward to talk more about functional GI disorders, why they have been involving mainly the abdomen. So microbiota has a lot of role in this. One of these is irritable bowel syndrome, as you know, and IBS can be in 3 different forms. One of them is predominantly associated with constipation. The other one is associated with diarrhea, and there is a mixed form as well. To understand dysbiosis, I'm going to share with you the Gordion knot. It's a real knot. We don't know how IBS occurs and what are the factors taking role in IBS patients. These include the diet, type of delivery, antibiotics, aging, inflammation, but also microbiota in the middle. So how are we going to solve this problem? It's an issue, really. So the knot, the Gordion knot, is a very famous knot, which is one of the toughest and well presented by Alexander the Great, who has given an example how to solve the tough problems and the knots. So we have been looking for a sword like Alexander the Great to solve this problem. Can it be probiotics? We are going to see more. So in the pathophysiology of IBS, the changes in the gut microbiota, in addition to, of course, the others, I can give you several examples. About the role of the microbiota and how microbiota changes the management of IBS. Of course, lifestyle modifications, stress management, the diet, these are important, but the probiotics have been shown that changing the rules of the game, I would say. So they are beneficial in recurrent abdominal pain, and they have role in the management of this pain. Let's focus on colonic motility a little bit, because inflammation in the colon leads this motility of the gut and the mainly large bowel. So inflammation led by microbiota can be managed by probiotics. And it has okay. Now, the colonic functions, let's focus on that a little bit. Colonic functions are driven by the luminal content, and the 50% of the luminal content done by gas and bacteria. So the stool formed by these unabsorbed food, carbohydrates, starches, and non-starch polysaccharides, and MACs, which are microbiota accessible carbohydrates, when they reach to the distal colon, provide us gas in addition to short-chain fatty acids. Let me summarize you the colonic motility a little bit and where microbiota takes a role. So the stool mass approaching to the distal colon triggers some stimulation, which will be transmitted to the central nervous system via spinal cord, and the central nervous system stimulates colon as a response, and the stool comes out. So what is the role of microbiota in this case? Yes, microbiota produce a lot of neuroimmune mediators, and translocation of certain mediators such as metabolic substances and short-chain fatty acids may directly alter and affect central nervous system. So this magical substance, short-chain fatty acid, is important, and they are produced by our microbiota. That's why probiotics are used in the management of constipation, and there are many studies I will be sharing with you very, very briefly, and some societies already decided to use probiotics in the treatment of functional constipation, such as Mexican society. Microbiota has a lot of effect on the transit time of the stool and the bowel movements. Well, I will try to show you changes in the microbiota in different age groups by these Spanish balls. In elderly, of course, microbiota is quite different than the infantile flora microbiota. So intestinal dysbiosis leads constipation. And here we see that some bugs such as Lactobacillus, Bifidobacterium, they are decreased in case of constipation, and some of them increase, such as Pseudomonas and Campylobacter. In IBS and constipation, there are some species and some population of bacteria significantly decreased. So gut microbiota really has a lot of effect on the colonic motility, as we see. And there are many more trials and publications regarding that. Even some news, such as the Harvard Health Publishing, suggesting us that the probiotics may ease constipation. I know some of these results are controversial, but I'm going to share with you a meta-analysis as well. We don't have time to go in detail, but the meta-analysis suggesting us that, yes, there is a place for probiotics in the management of colonic motility. So the probiotics are not only used for the GI system, they have a lot of effects on brain, on the liver. I will touch very briefly. So that's why we have to modulate microbiota. And these are the methods, such as management of associated health issues, such as balancing the diet, such as using human milk, which is really very important. That's why we have been trying to find a product containing probiotics, prebiotics, and mimicking human milk. But human milk is good for human beings. On the other hand, human milk may kill the other animals. In the modulation of microbiota, there are some other tools we use, such as antibiotics, probiotics, symbiotics, in addition to fecal microbiota transplantation. So it's helpful to correct dysbiosis, of course. Okay, let me touch a little bit to fermentation, because fermentation initially done in Göbekli Tepe 14,000 years ago, and we know that similar procedures have been done in Indus Valley of Asia, and then China and Japan, and which has been scientifically described by Elimechnikov, of course. So the prebiotic fibers following fermentation, mainly fructooligosaccharides and galactooligosaccharides, in addition to inulin, stimulates proliferation of beneficial bacteria such as Bifidobacterium and Lactobacilli. So they are good to provide us a balance, good balance, which is EUBIOSIS. So these are the ways how to modulate gut microbiota. But among these, today's topic is the importance of the probiotics. So probiotics, I don't have to define you again, are very well defined, but in practice, unfortunately, this definition very frequently forgotten. So some of the bugs are good, or they are helpful in the treatment or prevention of certain diseases, such as antibiotic treatment of Helicobacter pylori by antibiotics. So these probiotics are good in case of H. pylori related gastritis. There are many studies. One of them is regarding Bacillus clausi, which is really helpful to decrease some side effects of antibiotic treatment. So there are meta-analyses suggesting us that probiotics, in addition to eradication therapy, may provide us better success rate. So during the antibiotic treatment, maybe we will be in need of certain probiotics. So by killing the pathogens, probably we are in need of something to improve the good bacteria, which is not an easy procedure, because following antibiotic treatment and following the end of antibiotic treatment, eubiosis may take, to reach to eubiosis may take 5-6 months. In liver disease, of course, probiotics have several roles, of course, because liver disease can be associated with increased enterobacteria and decreased bifidobacterium, which leads severe cell necrosis. And at this point, probiotics, in addition to prebiotics, can be beneficial. So the question is, how can we achieve the beneficial microbiota? Of course, we need human milk for this purpose, which is not always available. And in this case, we need probiotics. So these are the main indications of probiotics, which may provide us eubiosis. I'm not going to talk or count all of them, but at the end of my speech, I would like to emphasize that slow changes occur in gut microbiota. So the alterations of early bacterial gut pattern may take months, even years, and these long-lasting functional effects eventually cause disease risk later in life. So probably we are not going to face some problems in early phase of our life, but in later phases, we will understand the importance of this healthy microbiota. That's why maybe some other new probiotics, new bugs have been described, such as Akkermansia muciniphila. This is another promising probiotic, and we are hoping to achieve good combinations of some other bugs to provide us eubiosis. So this is another interesting study recently published regarding Bifidobacterium animalis, which potentially we say improves the mucosal integrity. So, dear audience, at the end, I would like to summarize that dysbiosis leads disease-associated microbiome, and then it has to be remodulated. And we need something to remodulate. It's not always fecal microbiota transplantation, of course. At the first step, we need prebiotics and probiotics, non-invasive methods, by changing, of course, our lifestyle, by giving breast milk in case of infancy. We have to think about the type of the delivery. We need good genetic background, of course, the pollution, pathogens, management of stress, and the drugs, they all are very important. If we can remodulate them, we may reach to healthy microbiota. I think I have to stop at this point, and I'll be happy to discuss with you during the closing session. Thank you.
Thank you, Professor, for your wonderful presentation. So next panelist for the webinar is Dr. Metehan Ojen. He is a pediatric infectious disease specialist at Acıbadem University School of Medicine, Turkey. And after graduating medical faculty, he completed pediatric residency in 2000 and pediatric infectious disease residency in 2004. He served 5 years each in Inonu University and Suleiman Demirel University. At the moment, he is working in Eciba Dem University in Istanbul since 2015. He concentrated on microbiota studies for the last 10 years. He was the editor of first Turkish book and the editor of the journal Probiotic Prebiotic Chronicles for 4 years. He co-organized the first meeting of International Symposium of Probiotic Prebiotic in Pediatrics in Istanbul and second in Antalya. He is now the General Secretary of World Society of Prebiotics in Pediatrics, organizing the 8th Congress in Spain. He has more than 150 international national articles. He has performed more than 700 presentations on pediatrics, vaccines, microbe, infectious diseases, and ornithology in various meetings. He is also currently president of Turkish Pediatric Prebiotic Society. He has more than 2,400 citations and Google Scholar Edge Index is 25. Over to you, Professor.
Thank you, Bidhata, for the nice presentation. I think you see my screen. It's moving at the moment. And after all these 3 successful presentations, it's easier for me to talk about the microbiota and disease link, because I will talk about something related with the clinic, with especially for doctors or pharmacists seeing patients. As you see, this is for the last 2 decades after the millennium. The topic, the microbiota, is the most studied and will be one of the most studied in the future. The numbers are increasing, and when you see the PubMed, there is a new application here. You see, when you write microbiome PubMed, you see the numbers are rocketing here. So nearly 20 or 30 years ago, there were only a few articles or studies, but now it's rocketing and it is one of the most studied subjects. So we cannot even catch what is going on. But because of the many congresses, many these activities, we are really informed of very nice studies. So we just published this week this article by some experts all over the world, and we published it in the expert review of clinical immunology. In the beginning, we all believed that this dysbiosis, EUBIOSIS, or the function of the microbiota was related with the gastrointestinal system. So when you talk about probiotics, everybody thinks that it is about diarrhea, acute gastroenteritis. But after we discovered that this microbiota talk with your immune system, there's a crosstalk, and as you see, all these microbes, when they touch your gastrointestinal or respiratory epithelium, there are some anti-inflammatory or antimicrobial peptides. They are secreted to the lumen. But when they talk to the mucosa and submucosal lymphocyte, I mean the immune cells, as you see, there are many cytokines, thousands of cytokines, proteins, cells, antibodies. So this is secreted into the bloodstream. So it is not only the gastrointestinal system, digestion or constipation. Now we understand that dysbiosis, as all my previous presenters talk about, it is also related with all the autoimmune, cancer, allergy, and some type of infection diseases. So we understand that apart from the gastroenteritis, we can also cure some more diseases not related with gastrointestinal system. As you see on the right-hand side, these are generally related with the gastrointestinal system. But when you look up, you see there's a really close relation with anxiety, depression, some autoimmune disease like MS is very popular, also some skin disorders. This is one of the most common disease and will be in the future, obesity, metabolic syndrome. And even with vaccine response, yes, it is right. If you use probiotics, the correct probiotic during the vaccination, your antibody titers are increasing. Today, I will talk about the respiratory tract infection because I'm a pediatrist and you are a pharmacist and My wife is also a pharmacist. We have a pharmacy. I know very well that if you are taking care of patients, one of the most frequent complaint will be fever, cough, running nose. I mean the upper respiratory tract infection. So this is one of the biggest burden for the public system. There's a very good data about from USA. You see each year more than $40 billion are spent for the upper respiratory tract viral infection. Indirect cost is most of them. Even in the best medical system, even in the best country, 1/3 of these viral infections receive antibiotics. Unfortunately, there is a cost, but my concern is not cost. My concern is, as you hear, dysbiosis, because dysbiosis causes many autoimmune, cancer, and allergic disorders. But if you're a pediatric infection disease professor like me, working in a university hospital, we are very, very afraid of antibiotic resistance, because now antibiotic resistance is increasing because of this viral respiratory tract infection antibiotic load. And the WHO released and announces that even the minimal reduction, 5 or 10% antibiotic reduction, has paramount importance for the humankind. It is very important. Again, there is a very good data from CDC. You know, the regulation is very strict in the US. They study so much to tell the people to use the correct antibiotics. But as you see, for acute respiratory condition that are viral, the total for all ages, 50% of antibiotics are unnecessary. This is an amazing number. Can you see? All the antibiotics we use in the outpatient clinics, the half of them are unnecessary. So the WHO is now telling that the world is running out antibiotics. There is no new antibiotics. The resistance is increasing, and it is a global threat. And within 30 years, when it is the year 2050, my friends, the people dying from antimicrobial resistance, as you see here, antimicrobial resistance by 2050, it will be more than cancer again. So like it is the Stone Age, we will lose more people because of infection, because of resistant infection than any other. All my professors talk about the factors impacting the gut microbiome dysbiosis. As you see, the dysbiosis causes increased inflammatory cytokines. I showed the figure in the beginning, and it is released to the blood and the lymphatic system, and it goes to the target tissue or organ, and wherever it goes, as you see, there are many allergic and autoimmune diseases or disorders we have. So it is more important if it is in the first 1,000 days. I mean, if the child is small, the impact is more because the richness is decreased, the immune system is not same. But as you see, if it is during the first 2 or 3 years, the impact is very increased. So this is a wonderful figure. After the Second World War, there was the Industrial Revolution. We believe that everything will be okay. But because of this drugs, especially the antibiotics, of course, antibiotics saved thousands, millions of lives. But now more than half of the antibiotics used for the viral infection. And the industry, which is represented by refrigeration, because it symbolizes that we use many unnecessarily food with additives, unfortunately. And as you see, within the last 50 years, there is again rocketing numbers of asthma, food allergy, atopic dermatitis. So that's why we have to support our immune system, because we have highest number of C-sections, highest number of antibiotics, because of this refrigerators or not organic or natural food. So we need an immune system support. But whenever you see, whatever you read, whatever it is sold in the pharmacy, they always claim that they are immune boosting. So this is not the true, as you know. It is easy to claim, but it is difficult to prove it. So there are some products that have been studied in the humankind. So we should look at these products. One of them is probiotics. So this was the first systematic review we published in 2015. So until the 2015, of course, we summarized the pediatric age group. We excluded the infant formulas. So we used the high quality with double-blind randomized control trials. And these are our tables. There are 14 studies. These are the results. The only thing important here is that, as you see, when you check the outcomes, they have used different outcomes, such as this study look for incidence of fever, this look for the incidence of infection, number of antibiotics, blah, blah. So each study look for different outcome. That is why we didn't run a meta-analysis. So we just reviewed. We had a summary of the literature. If it was a meta-analysis, then it will be a solid data and it will be placed in the guidelines. This systematic review shows us that these 14 studies, about 6,000 children, most of them before the primary school, preschool children. As you see, 12 of the studies reported for incidence of infection, respiratory tract infection, and 7 had significant decrease in the number of infection, and again 7 had significant decrease in the severity of symptoms. So this means if you use probiotics prophylactically, I mean, if you use for 3 or 4 months during the winter, the number of infection is decreasing. Plus, even if you have an infection, the symptoms, the severity of symptoms is decreased. So this is very good because if you have milder symptoms, so you do not rush for the doctors. So you doctors do not write you, prescribe your antibiotics. So as you see, in half of the studies, the incidence of infection and severity of symptoms decrease, and it is similar for the 8 studies just reported the missed days of schools. As you see, half of them had significant decrease. So this shows clearly that not every probiotic is probiotic effect on respiratory tract infection, but if you know the correct strain, correct probiotic, and if you use it for 3 or 4 months, during the winter time when the schools are open, it shows that the correct probiotic decreases your number of infection and your severity of symptoms. And 5 years later, it was such a hot topic that in 5 years, there were 20 more studies. So we again re-summarized the literature 2 years ago. So in this news review, there were 33 high-quality studies, randomized double-blind, more than 11,000 children. Again, most of the children were preschool age, but now there were some infant. This is very important because the infection rate is low under the age of 1. So they have even checked under the age of 1. As you see here, of these 33 studies, 30 studies reported the number of infection and 20 had significant decrease. And for the severity of symptoms, 20 reported and 10 had. Nearly half of them had impact on the severity of symptoms. Again, the missed day of school and incidence of antibiotic use is just reduced statistically in half of the studies. So again, we documented that if you use the correct strain, if you know which probiotic is good for diarrhea, which probiotic is good for respiratory tract, which one is good for allergy, so it will be good to advise to your patients, to your if you're a pharmacist visit, I know because many people coming to the pharmacy first before going to the doctors. So you can recommend these probiotics for diarrhea, acute gastroenteritis, these other probiotics for respiratory tract infections. So this is not only for the pediatric case. As you see, there is also adult study. Here they had 7 studies to nearly 2,000 people. They show that if you use 3 to 8 months of probiotics, with probiotics, only 20 of the 100 patients have infection, but if you do not use 30 of the 100, so it shows 1/3 decrease from 30 to 19. It shows that 1/3 decrease. And for the number of infection, as you see, if you do not use probiotics in a winter time, you have average 4 infection upper respiratory. If you use probiotics, you have 3 infection, 1 less infection. And the duration of the fever, duration of the infection is 2 days less. So this is very, very important, my friends. The severity of symptoms, the duration of the infection is 2 days less. So this is very important. So you can return your daily life, your school, your work 2 days before. And as you see, the number of people prescribed antibiotic is really from 20 to 30. So it is something like 35%. Again, 1/3 is decreases. Don't forget, WHO recommends 10% decrease will be very important for the humankind. So there will be 1/3 decrease, 35% decrease. So we should know which probiotics are good for preventing upper respiratory tract infection. And decreasing antibiotic is very important. So we have one of the longest article in the literature. So we all summarize the adult, the McFarland, one of the guru of this subject. She summarized the adult antibiotic-associated diarrhea, and we summarized with my partner the pediatric one. But I want to show you that if you again use the correct probiotic, it prevents antibiotic-associated diarrhea, as you see in the meta-analysis. Because now the primary outcome is similar, so we ran a meta-analysis, and it also prevents the Clostridium difficile infection. But what we want to show here is, you see the red line. So if you do not use probiotic with antibiotic, you have this much dysbiosis. If you use probiotic intervention during antibiotic, as you see, there are two options. One, the dysbiosis depth may be milder, or the dysbiosis time may be shorter. So in any way, if you use one of these studies, one of documented probiotics with antibiotic, in the beginning, we believe that 2 weeks is enough, but as the studies increase, we now know that dysbiosis continue for 4 months, 6 months. So you can advise your patients 3 months, 4 months, something like that. If they use probiotics, dysbiosis resolve. So there's a remodulation sooner. And finally, as you see, this is the recommendation from the masters, the Martin Floch and the Alan Walker. The first two names are the professors who invented this mucosal immunity. They spent for a lifetime. They also invited us for the new guideline in 2018. So we were in the new guideline, but then there was COVID, as you know, so the new guideline is not published, and Mary Ellen Sanders will, I think, will publish. Here you see there are recommendations that some strains are very effective against the infections, so they support your immune system. Some strains for allergies, some other strains for diarrhea. So you should know which strains are good for which disease, so you can recommend your patients these probiotics. And finally, antibiotic resistance is the major problem for humankind because there is no new antibiotic. We should not use antibiotics for viral infection. So if you know that some strains, certain strains, if you use them regularly for a few months during the winter time, it decreases the number of infection, and this, even if you have an infection, the severity of symptoms. Of course, as always, we need more studies, more multi-center studies, looking for the same outcome so that we can run a meta-analysis. And there are some new topical applications of this oral probiotics with topical applications like puffs. These are very promising. So we are now organizing the 8th Congress in Valencia in September. So if you are available, so it will be hybrid congress, both face-to-face and online. So if you are available, we are waiting for you. Or if you cannot come to Spain, you can email me, or I can direct you to the agency. We can give you a discount price for online registration. So please be my guest to listen 3 days of this wonderful academicians, the new studies. Thank you so much for your patience, my friends.
Thank you for your presentation, Professor. Finally, we have reached the Q&A session. So in this session, I request if anyone has question, they can put it in the Q&A box. And for now, we have received a couple of questions from the attendees. So the first question is relationship between gut wellness and asthma. Any of the panelists would like to answer? I would like to request to on their video.
May I?
Sure.
Yes, as I show in my slide, now more than 150 autoimmune, allergic, and some kind of diseases, they have shown that there is a relation with dysbiosis and these disorders. Of course, the most important is dysbiosis causing asthma or asthmatic patient have dysbiosis? So both of them, both ways are correct. So dysbiosis plus genetic susceptibility. So if you have susceptibility, hereditary predilection, and plus you have a epigenetic dysbiosis like cesarean section, antibiotic in the first 1,000 days, and this environmental changes. It may increase your chance for asthma, and after asthma you use many drugs, antibiotics, and again, so you have more dysbiosis. So there is a vicious circle. This is true for many autoimmune diseases.
Thank you for the explanation, Professor. We have other question, which is regarding microbiota. If microbiota can cause anxiety or depression. So if anyone would like to answer the question. Professor Ender, would you like to answer the question? Can microbiota, sorry,
I heard the question. This is a very interesting question, since there is the interaction between the brain, the central nervous system, and the others. Microbiota related changes such as, briefly, dysbiosis may lead alterations of mood pattern as well. So there are handful of studies suggesting us that the mood is affected by microbiota. In case of depression, in case of neurological problems, there are changes in microbiota composition. But we need further studies to prove which kind of microbiota can correct dysbiosis and dysbiosis-related mood problems and neurological problems. We are not at that point so far. But happiness, sadness, depression, yes, these are linked to microbiota changes.
Thank you for the explanation. We have other question also in the Q&A box. And the question is, what is the best probiotic to take during pregnancy? So, Anna, would you like to answer, if you know the answer?
There is no best probiotic to take during pregnancy. So every continent, every country, every culture, every humidity, every environment has a different microbiota. So it is different in all parts of the world. So there is no simple answer for this one. It is not such a simple question or simple answer. So it depends on the woman, country. So there is no answer for this one.
Maybe we have to comment on this, like Dr. Rosen, he's right. During the pregnancy, why do we need probiotics? Do we think that the probiotics ingested by mouth eventually change the composition of microbiota during intrauterine life? We have no study like that. And on the other hand, some microbiotas can be dangerous during pregnancy. We don't have sufficient evidence regarding the safety of these products. So we have to be careful to give message that during pregnancy, if you can ingest lactobacilli or more other beneficial types, or even very popular Akkermansia, can we prevent further dysbiotic illnesses? We don't have sufficient scientific evidence for that so far.
Thank you for your explanation. We have some other questions as well. The other question is, which specific probiotics could be recommended to use in Latin America?
I guess this is, it goes in the the same answer that Dr. Mehta has given us. We don't have a specific probiotic for a different, a specific kind of person or ethnics or whatever.
Dear Anna, but the beginning of the question was different. It is known that, and Norma Murula is asking for diarrhea, acute gastroenteritis. So they say that every 9 children treated, probiotics prevent 1 case of diarrhea. There is no such a data because we have done and published more than 10 gastroenteritis study. We do not prevent diarrhea, we treat diarrhea, and what we treat is we try to shorten the diarrhea period. So if you start the correct probiotic, which is also available for Latin America guideline, because it was also published by Latin America Pediatric Gastroenterology Group. So if you use the correct probiotic, it decreases 1 day, nearly 24 to 28 hours. So it shortens the diarrhea. So we treat the diarrhea. I think the friends try to ask the number needed to treat. So number needed to treat is something different. Prevention is something very different. So there is no relation between this data. What I try to say is if you use the correct probiotic, which is available in the Latin America guideline, you shorten the diarrhea for 1 day.
Thank you for the explanation. We have received other questions as well. And so the next question is, as a pharmacist, the recommendation of a probiotic supplement, can it be useful for diabetic and GI problem patients or even patients with insomnia problems? Anyone would like to answer the question?
Yes, my sir, Ender Pehlivan is also my professor. He educated me so much.
Dr. Özen knows much better this kind of pharmaceutical approaches. So I would like to leave the answer of the question to Metahan, who is an expert, who is a leading expert of this field.
Thank you, my sir. But these questions are very general questions. So we are studying so specific. So you have listened to studies from the Anna Faria. Their studies are very specific. The outcome is very specific. So can be useful for diabetic patients. Certainly, there are many probiotic studies showing that it decreases the insulin resistance. It is good for the GI problems. But when you say GI problem, there are thousands of GI problems. So for some GI problems, it is very useful. But for some of them, it is not useful. So you should be very specific about the problem, the people, the country. There are some studies that it is very good in Japan, but the same strain is not good in Spain. So it depends on the studies. So we always talk about we need more studies, more studies.
Well, for the part of insomnia, if I have heard correctly, there was a part for insomnia.
Yes.
Yes.
Okay, I can
Yes.
comment on that because we have an experience. Because last year we have done a study among university students, the relationship between insomnia, sleep disturbances, and the diet, as well as their usage of probiotics. We have found that in the student group, ingesting MACs, I mean microbiota associated carbohydrates, the sleep problems were less. So this is a kind of preliminary result of our study at the university, but it implies that the students are consuming less and less fiber and less MACs in their diet. If we can increase the amount in their diet, that may help insomnia as well. This is quite preliminary result, by the way.
Thank you for the explanation. We have some other questions as well. I hope this is the second last question for today. Which artificial probiotics are recommended to take in daily life? So this is the next question.
Dr. Metehan is the right expert. Ask him, please.
There is no answer for this, sir.
Okay, then last question for today is, in your personal opinion, it is necessary for older adults to consume probiotics on a regular basis? I think it's a question. Is it necessary for older adults?
So my opinion is, if somebody has a kind of dysbiosis, they should use not only probiotics, prebiotics, symbiotic, postbiotic, fermented good, and move from the city to the rural area, have a dog, have a cat, walk on the grass and have some sea or something like that. Because whatever you do, there are unbelievable number of studies from the northern part of the world, from Scandinavia and Canada. If you even do everything right, if you have a stress, that we have too many stress nowadays, you have a dysbiosis and the number of autoimmune diseases are increasing. So having a probiotic is not the only solution. It is not that simple. We are a very complex individuals. So you should do many things together. You should have a good family life, a good business. You should eat good fermented food. You should have, of course, some probiotics, prebiotics, fermented good, go outside, have a dog, walk out. So having one probiotic will not be a solution only.
Thank you for the explanation. I think with this, we wrap up the Q&A round. And I think we have come to the almost the end of the today's session. So as a conclusion, I hope that this session was useful. And with this webinar, we have understood more about the gut wellness, probiotics use, and related things. And we also take this opportunity to invite you to the 80th FIP World Congress of Pharmacy and Pharmaceutical Sciences, which is happening in Seville, Spain from 18 to 22 September 2022. And the theme is, sorry, can you just go back? I was about to tell about the theme. The theme is Pharmacy United in the Recovering of the Healthcare. And there are several topics for the FIP World Congress. And you can visit the official website, seville2022.fip.org for details about the Congress. And you can check all the future FIP digital events at events.fip.org. And I would take this opportunity to thank everyone for attending this event and making it successful, especially all the speakers despite their busy schedule, and even the attendees. Thank you.
Thank you.